In this lecture, we will introduce fats and lipids as our second macronutrient. So again, we saw this slide in the carbohydrate lecture, just overview of the macronutrients. So remembering carbohydrates, fats, and proteins, larger molecules made up of monomers, all whole naturally occurring foods will contain some combination of the three macronutrients. And in the body, we see macronutrients occurring not necessarily in isolation from one another, but oftentimes bound to each other to form various molecules. Some examples being the glycoproteins, glycolipids, and lipoproteins. As we'll see in this lecture, one of the primary functions of fats, as with carbohydrate, is to provide energy. And again, in the third lecture, we will look at proteins. So just a little background on fats and lipids. So this term lipid is often used synonymously with fats. For our case, we're going to use lipid to refer to those types of fats that are liquid at room temperature. And again, for our purposes, when we talk about fats, we will typically refer to fats that are solid at room temperature. But do also know that I may actually interchange the words. And oftentimes in the scientific literature, you'll see them used synonymously. So in general, and I'll say this too, fats is often used as an umbrella term to include both solid fats and liquid fats or lipids. So fats generally are not soluble in water. The term we use there is hydrophobic, so phobia, afraid of hydro water. And this it becomes important to know when we start to realize that our blood is water-based, and so most of our body is water-based. And so when we are ingesting fats and needing to utilize especially essential fats, or when we ingest things like fat-soluble vitamins or other molecules that might be fat-soluble, how do we then integrate them into the body? How do we even digest them? So we'll see that in some of the following slides. We are also going to go through three classifications of fats. We're going to look first at the triglyceride, then the phospholipid, then the sterol. Remember, just like with carbohydrates, fats are composed exclusively of these three elements, carbon, hydrogen, and oxygen. Remember, too, that protein is composed of these three elements and also nitrogen. Some of the primary functions of fats. So you'll see that energy is sort of the last thing I list here. Fats are the structure for a lot of our hormones. They're also the structure of our cell membrane. They also work in various cell functions, primarily in cell signaling, but also cell fluidity and the ability of cells to bring in molecules and send out waste molecules. And then, as we know, we can use fat as a source of energy. It's typically we prefer it second to glucose. And most of us, I think, are familiar with the notion that humans can actually store fat on the body. And when we refer to fat stored on the human body, we refer to it as adipose tissue. So that's technically stored energy in the form of fat. Recall that when we store glucose as energy, we call that glycogen, and glycogen is stored pretty exclusively in the liver and in the muscles. Adipose, we can basically build adipose tissue just about anywhere in the body. We can even build some fat storage in between muscle fibers. So let's look first at the triglyceride. 95% of the foods that we eat are made up of triglycerides, and we'll look at different types of triglycerides. Also know that a majority of the fat that exists within our body is triglyceride. I shouldn't say within our body, but when we measure fat circulating in the blood, we're typically measuring triglycerides. We may also measure some cholesterol molecules, but the majority of the fat that we'll find in the blood would be triglyceride. And definitely when we store fat in adipose, we store it as triglyceride. The triglyceride does have monomers, just like with carbohydrate. In the triglyceride, rather than having one singular type of monomer that might take on a few different shapes, the triglyceride has very specific monomers. So glycerol being the backbone, if you see this 3-carbon chain, running vertically, this is glycerol. And then the 3 fatty acids are these carbon-carbon chains that attach horizontally to the glycerol molecule. So triglyceride is 3 fatty acids in a glycerol molecule. Those are the molecules. So let's look at how we classify triglycerides then. Basically, there are 2 ways to do this. We can classify triglycerides by the saturation of the fatty acid chain. We'll look at that definition in a second. And then we can also classify triglycerides by the length of that fatty acid chain. Here's saturation. If you recall from basic chemistry, carbon has 4 available binding sites. So in a carbon-carbon chain with hydrogens bound to any of the available binding sites on each carbon, we see that in a saturated fatty acid molecule, each carbon is bound maximally to hydrogen. To say that in another way, we have this base chain of carbons. So therefore, every carbon at least in the middle of the chain is going to bind on one side to a carbon and on the other side to a carbon. So that's 2 out of 4 binding sites carbon is using up. That means there are 2 remaining binding sites. And so to be saturated, technically being saturated with hydrogen in this case, the carbon then binds a hydrogen in either of the 2 remaining binding sites. So in this chain, we see that every single carbon in the middle of the chain is bound to another carbon behind it and in front of it, and then bound to 1 carbon on, above it and below it. Or sorry, 1 hydrogen above it and below it. So we call this a saturated fatty acid chain because all of these carbons are saturated in their binding capacity. They're bound to as many molecules as possible. When we then look at unsaturated fatty acids, we notice one pretty major obvious change. So we see the insertion of a double bond. Again, kind of basic chemistry, these carbons can bind to each other not just by sharing 1 electron, but they could actually share 2 electrons. So in this case, the double bond occurs because the carbons are sharing 2 electrons, which then means they're still bound. Each is bound on one side to another carbon. But then because they share 2 between them, now that's 3 binding sites that are used up for only 2 bonds. So then there's only 1 remaining binding site to bind 1 hydrogen apiece. So you notice there's an empty, technically this isn't empty because the bond is over here, but we know that this is not saturated or it's unsaturated because these 2 carbons now are no longer bound to as many other elements as possible. So this is an unsaturated or double bond. And you see over here I say shape. So when we talk about saturation of the fatty acid chain, when we see the double bond existing, you notice how this now has what we call a kink. It's bent. So the presence of a double bond in a fatty acid chain typically determines the shape of the fatty acid. So unsaturated fatty acids are going to be a little bit more fluid in nature, again, lipid, and saturated fatty acids are going to be a bit more solid in nature, or fat solid. Here's another schematic of that. So these lauric, myristic, palmitic, and stearic are 4 types of saturated fats that exist in our food supply. So you see all of them have a pretty straight linear chain. On the right side, we see oleic, linoleic, alpha-linoleic, gamma-linoleic, and ricinoleic acid. You notice some of these are monounsaturated, polyunsaturated, poly-mono. But what you notice is that they're kinked. So where the double bond occurs, the chain now kinks. And again, just like in monosaccharide and polysaccharide, we know that mono means 1 and poly means many. So in a monounsaturated fatty acid, we have the presence of just 1 double bond. In a polyunsaturated fatty acid, we have more than 1 or a few. Obviously, the more bonds, the more kinky it becomes. But you could see if you tried to stack up a bunch of polyunsaturated fatty acids, it wouldn't go so well, hence a liquid. If we did try to stack up a bunch of saturated, they would all fall into place pretty nicely, and hence. Here's one last schematic of that. Again, saturated fatty acids typically taking on a solid shape in food, and unsaturated fatty acids typically taking on a liquid in foods. Also important to note that most foods, just like they contain a combination of our macronutrients, most foods contain a combination of different saturated and unsaturated fatty acids. So again, we kind of classify the food as being a saturated or an unsaturated fat based on what it has the most of. So most foods that take on a liquid shape at room temperature are primarily unsaturated fatty acids. Most foods that take on a solid at room temperature are primarily saturated fatty acids. So next we move to fatty acid chain length. This is really straightforward, but you'll notice we have three examples of fatty acid: butyric, acrylic, and palmitic. You don't need to know these names, but just understand there are lots of different types of fatty acids in our food supply. So butyric is an example of a short chain fatty acid. You notice that in the chain, there's only 4 carbons, so pretty short chain. Medium chain fatty acid, acrylic as an example. This one has, I think, 8 carbons, maybe 10. I didn't count. But medium chain fatty acids being 6 to 10 carbons in length, medium chain. And then palmitic acid is an example of a long chain fatty acid containing 12 or more carbons in length. You see at the bottom I circled digestion absorption. Again, pretty intuitive. The longer the chain, the longer it takes to digest. Shorter the chain, the faster it is to digest. Okay, I knew I put these slides backwards. So this is going back to saturation. Forgive me. This is going back to saturation of the chain. So here again, we see saturated fat. We have a straight chain. Unsaturated fat, we have a kinked chain. And I need to introduce this concept of a trans fat. So trans double bond. A couple of things that are important to note. Trans fats are rarely, if ever, found in nature. Generally, we say that they're not found in nature, but we are not the omniscient beings we think we are, so it's possible that maybe trans fats occur in nature. But generally speaking, trans fats are man-made fats. So what we've done here, if you notice, if we go up to the unsaturated fat, the linoleic acid, you notice the carbon-carbon double bond, and we call this a cis bond. What that means is that the carbons are in the same orientation in space. So we are looking at this as a 2-dimensional molecule, but fatty acids are 3-dimensional molecules. So if we looked at this as more of a 3D molecule, we'd see that these 2 carbons are in the same orientation in space, and hence their singular hydrogen bonds go off in the same direction. So we call that cis or same. In the trans fat, we notice that at the site of the carbon-carbon double bond, the remaining attached hydrogens to those carbons are in opposite planes in space. So trans being opposite. Again, not something we see commonly in nature. So what we've done here is we've applied some type of hydrogenation where we sort of force this bond to take on a different orientation in space, so that it goes from being in this cis shape, which causes a kink in the chain. So when they're on the same side, you can kind of imagine it causes the chain to bow out or bend in the opposite direction from where the hydrogens are. And if we swap hydrogen bond for one of these carbons to force it to bond on the opposite side, we see that, thus creating a trans double bond, we see that actually sort of takes the kink out of the chain. So we did literally do this around the time that we created margarine. We wanted something that was solid in nature, and we were using a lot of vegetable oils because we thought those were healthier than regular butter, but we also wanted butter. We were trying to eat less butter. So food companies figured out how to hydrogenate and shift the shape of this double bond from cis to trans, thus making a solid fat out of what was originally linoleic acid, a liquid fat. All right, so that's triglycerides. Moving on to phospholipids. So phospholipids are really cool. They are, as the name would imply, it's a combination of some sort of phosphorus molecule with the lipid. So in this case, we have we have two fatty acid chains in a phospholipid. We call that the hydrophobic tail because we know fatty acids are afraid of or don't bind with water. And we have a hydrophilic head. Usually that contains the molecule phosphorus, but the hydrophilic head will vary depending on what the molecule is. I should say, sorry, it contains the element phosphorus. So functions of phospholipids, transportation in the blood, particularly important in our case to talk about fat-soluble vitamins, but also to transport fats in the blood. We use phospholipids because they have one part that is philic or loves water, and one part that is phobic or afraid of water, hydrophobic, hydrophilic. So the hydrophilic part can sort of float or bind in the water, the blood, and the hydrophobic tail can sort of, or I should say philic can kind of bind to the blood and move in the blood, and the hydrophobic tail kind of floats on top. We will also see phospholipids in the digestion of fat or bile. When we talk about the digestive system in a couple of weeks, we'll learn that bile is a molecule that helps to break down or emulsify fats. So in order to do that and cause, just like with soap, in order to allow the fats we've taken in to break down and bind with water, we need to have some sort of hydrophobic and hydrophilic molecule. We'll also see phospholipids existing in the cell membrane. You might remember the term phospholipid bilayer, and also knowing that these molecules assist in cell signaling or communication between cells. Here's your schematic of the phospholipid bilayer. The phospholipid, not the bilayer. So you see that you have two tails. We could have saturated or unsaturated fatty acids in the tail. They'll vary by molecule. And here you have the hydrophilic head. So still containing that glycerol molecule, but then some sort of other, some sort of phosphate-containing molecule at the top. Again, so you see this R chain up here. This sort of means that R is kind of the variable. So whatever the phosphate-containing molecule is, this R chain or whatever elements bind off of this will vary from phospholipid to phospholipid. Couple examples, again, we see the phospholipid bilayer. So we kind of start to recognize these molecules with this phosphate head and these two fatty acid chains. So if you remember, this is the structure of the membrane of every cell. And again, you'll notice the hydrophilic head is the phosphate head, and the fatty acids are hydrophobic. They face towards each other in the middle. So the lumen of the cell, this is the inside of the cell, and the extracellular space is outside of the cell. So interesting how the structure of every cell in our body is to put the fatty acid molecules towards each other. So every cell in our body is sort of surrounded by this fatty membrane that has to, that either end of the fatty acid membrane is these phosphate heads. And then here you see bile acid on the left side. So again, having the hydrophobic side and the hydrophilic side, and just sort of interesting how this is a really rough schematic of, in our digestion, what would start to take shape when we are ingesting lipids or fats. They'll be surrounded by these hydrophobic and hydrophilic bile acids and start to emulsify them or break them down so that eventually we can join them with other phospholipids in the blood and transport them. Okay, so that's triglyceride and phospholipid. And now our third one is the sterol. Sterol takes a slightly different structure from triglyceride or phospholipid. The sterol is like a multi-ring structure. So you see that base structure here with A, B, C, D. And on the next slide, I'll show you some further examples of sterols in the body, and they'll all have this basic structure. So on the far right, I have an example of cholesterol, and you see that it takes that A, B, C, D, the four-ring structure. So cholesterol is a type of sterol. It is abundant in animals, including humans. It's not found in plants. Plants don't make cholesterol. So humans do make cholesterol. We are animals, so we do make cholesterol. Virtually every cell in the body produces it. And so because of that, we say it's not an essential nutrient because we can make it, so we don't need to ingest it. There's no biological need to ingest cholesterol. A couple of really important functions of cholesterol. We always think of it, well, commonly these days we think of it as a not so good molecule, but actually it is a very important molecule. That's why our bodies make it. So cholesterol is just like phospholipids. It's an integral component of cell membranes. I do have a picture on the next slide, but the reason we have cholesterol in between, so the cholesterol molecules will sit in between the phospholipid molecules, and they help keep that cell membrane fluid, right? Like we don't want tough, rigid cells and cell membranes, right? That would probably really hurt if we were made up of these really tough, rigid cell membrane structures. So we want some fluidity. We want to be sort of like, soft, kind of, not super soft, not liquid cell membranes, but kind of a balance between soft and rigid. So that's actually what cholesterol helps with. And cholesterol actually helps with like electrical impulse transmission throughout the body. And cholesterol is also an important molecule in brain cells. And I'm not sure why this one is indented so far. This indentation should be back out here. I'll try to fix that. But this is really important, and this is what I'll show you on the next slide. Cholesterol is the basic molecule for reproductive hormones, for vitamin D, and also for bile salts. So bile being kind of made up of cholesterol and phospholipids. Here's your schematic. Here's your basic molecule of cholesterol. And here we see a lot of our reproductive hormones. So estradiol, testosterone, progesterone, aldosterone, and cortisol. You can see how they all have that 4-ring structure. They're all cholesterol molecules, basically. And then this is really interesting, I think, understanding how the body makes these various reproductive hormones. So again, always starting from the cholesterol molecule, and then we go to pregnenolone, which we kind of call the mother of all hormones. Pregnenolone can then make another hormone called DHEA, or a hormone called progesterone. From progesterone, we can make cortisol. We typically think of that as our stress hormone, but it's also sort of our awake hormone, right? It peaks in the early morning, it kind of has a lull in the mid-afternoon, and it peaks slightly again in the late afternoon, and then really drops down at nighttime so that we can sleep. Progesterone can alternatively make androstenedione, which is then kind of the starting place for our true reproductive hormones. So from androstenedione, or not true reproductive, but for our estrogens and our testosterone. So that can either go to make testosterone or estrone. Estrone can make estradiol or estriol, and these are we call these the 3 estrogen molecules. Okay, and then here's that schematic of cholesterol sitting in between the phospholipids in the cell membrane. So fluidity and stability of the cell membrane. Without these, the membrane would easily split apart. So super important structural. All right, and then kind of a recap, the functions of fats: energy storage, cell membrane and growth, and that's throughout the body. This is also really important too, so reversing DNA damage, immune response, and tissue repair. So it kind of makes sense now that we're starting to see the importance of triglycerides, phospholipids and sterols in the function of the cell membrane. So if we are reversing damage to tissue or repairing tissue, we need these types of fat molecules to help build new tissue. Transportation, absorption, and cell fluidity. We started to largely take fats out of, I guess I need to start over. Not too long ago, there was a lot of fear hype in the popular media about fats in our foods. So we started to take fats out of our food. But what we did in doing so, though, was we sort of lost a lot of really important qualities of food. So fortunately now most people are starting to realize that fats are really important in our food supply. And part of the reason for that is because they really make food is really satisfying, but also because we know fats are essential molecules, especially some of those unsaturated and unsaturated fatty acids. And we'll look at that in the next lecture. But fats in our food, lipids in our food provide texture. They are definitely the source for flavor, flavor beyond sugar and salt. But a lot of other flavor molecules are fat soluble. So any sort of compounds, molecules in our food that provide flavor have to be bound to fat. So if we take fat out, we don't taste their flavor. Also for mouthfeel, so just like texture and sort of like how how it feels in your mouth. It's not a flat food, right? It has like a really nice texture. It has really nice, feels really good as we're chewing it. And so again, that then relates to satiety and satiation. So like feeling really satisfied and feeling full after we're eating. I don't think I wrote this out, but it's in one of the, I think it was back in Unit 1, energy density, referring to the amount of calories per volume of food. So fats are the most energy dense of our foods, of our macronutrients. Proteins and carbohydrates provide 4 calories per gram of that molecule. Fats provide 9 calories per gram. So they are far and away more energy dense than either carbohydrate or protein. And then how much do we need? The acceptable macronutrient distribution range is about 20 to 25% of total daily calories. Again, these are rough estimations and vary person to person. Certainly we know to avoid trans and hydrogenated fats. These are the man-made ones. Including fats from plant foods gives us not only the essential fats, but also fiber, micronutrients, and these are mostly polyunsaturated. Understanding that when we consume fats from animal foods, these are mostly saturated fats, and in some cases, depending on the quality of the animal flesh, would provide very few, if any, other essential nutrients. And one last little schematic I'll let you kind of take your time to look this one over, but I think I gave this a pretty good run through, and I think this is definitely a good chart to follow. So kind of which saturated fats theoretically would be good for our bodies, which unsaturated fats would theoretically be good, which unsaturated fats to avoid, and which saturated fats to avoid. And again, most of these you'll see are trans fats. And to kind of help us understand these unsaturated fats a little better, we'll go to the lecture on essential fatty acids next.